We are delighted to have our next speaker, Dr. DeAnna Baker-Frost. She is part of our MUS team. It's really not been a long time, but it's been a big impact. So it feels very powerful, and we are delighted for her to share with us updates about research. Let me just give you a little bit of the background that I pulled off of her if you haven't met her before. So she's a native of Baltimore, Maryland, and she completed a combined MD and PhD program here at MUSC and an internal medicine residency at Duke. And then upon completing her residency, became a board certified internal medicine physician and returned to MUSC to our great favor. She is passionate about both patient care and research. This is part of what makes MUSC exceptional, is the blending of those clinical experiences with research questions and research initiatives to help advance treatment, management, hopefully one day a cure, or prevention, even better. But, Dr. DeAnna, as we say, is like our folks here at MUSC, quite generous with her time. And she is a supporter of what's called the Charleston support group. She helped get that launched and also continues to stay in touch with the group as it's evolved to the South Carolina group online. So she doesn't just see us in the office, right? She's seeing us in all different aspects of where we are. And this is invaluable. So Dr. DeAnna Baker-Frost, please share with us some updates. Thank you.
All right. Good morning, still morning. Good morning, everyone. Thank you very much, Karen, for that very kind introduction. So as you know, I'm DeAnna Baker-Frost. I'm one of the rheumatologists here. And my goal for today is to update everyone on the exciting times that we're in for scleroderma research. I'm very hopeful that we are moving closer to finding better treatments for our patients. And I'm hopeful also that this presentation will allow you, and hopefully you find Dr. Silver after the talk, as he said, at lunchtime, so that you can understand the importance it is now more than ever that we continue our research funding. Let's see. So excitingly, in the year of 2024, there was over 1,000 research manuscripts that was published about scleroderma. And we're not just talking about here in the United States, we're actually talking about internationally. And I'm proud to say that MUSC has been one of those contributors to updating the recent advances for scleroderma. So this is our little website here. It's called PubMed, and it's run by the National Institutes of Health. And all I did was a brief search and just put in the word scleroderma. And you can see there was 1,100 results that came about. And so those topics ranged in possible treatments, possible genetic causes to the reasons why people develop scleroderma, as well as bringing more awareness to the mental health needs of our patients. So the discussion today is not meant to be a complete summary. We don't have several weeks to sit here and go over every single publication, but I wanted to hopefully give you a sense of all of the different research areas that's being highlighted. So some of the topics that we're going to talk about today will cover possible treatments for inflammation. And these will include cell-based therapies, as well as other mechanisms to block inflammation and the inflammatory proteins called cytokines and how they signal inside of the cell. We'll also talk about pulmonary arterial hypertension. We'll talk about gut health. We'll talk about fibrosis, which is also a really major part of scleroderma, including the genes that are associated with fibrosis, as well as some possible inhibitors of fibrosis. And then finally, we'll talk about social determinants of health and mental health. So I wanted to start here because I think this will give us a good background on when we start our discussions about cell-based therapies. And I know that you guys are a very savvy audience because I get questions in my clinic regarding some of these cell-based therapies. And so I want to kind of highlight where it says the innate immune cell, T cells, and B cells, because those are the three cell types that are really interesting, really in an exciting time right now that could lead to some of the potential therapeutics, and it will kind of garner some of our conversations. So the innate immune cells is a part of the immune system, and they are very important for kind of calming down the immune system and fighting infections, just as T cells are and as B cells are. And B cells are the types of cells that are responsible for producing antibodies. Now, antibodies are helpful because it helps us to fight off infections when we are bombarded with them when we're out in the world. However, they also are responsible for producing autoantibodies, which are basically what those little Ys look like. Those are antibodies. And some of the autoantibodies that you may have heard about, that you may have had your clinicians talk to you about, include SCL70, centromere, as well as RNA polymerase 3. So those are the cells that produce those recognized autoantibodies in scleroderma. And so, as we mentioned, B cells, they produce the autoantibodies, but then they can also produce some of those inflammatory cytokines as well. And so inflammation is also a really big part of scleroderma. And so because of the importance of B cells in causing the disease to develop, there have been several researchers that's looked at cell-based methods to try to get rid of those B cells. And so some of these names will sound familiar to you, and they include the CAR T cells. Those includes the CAR NK cells. And then there are a different, even other cell type called the BITE cells. So we'll talk into a little bit of detail about what these therapies really are. So CAR T cells are probably the most known ones, but CAR NK cells and then BITE cells. So what is even a CAR T cell? You probably have seen and heard lots of different articles that's been presented either in press or in the news about these amazing cells. So CAR T cells is basically being able to get T cells out of the body and then manipulate those cells so then they can go and attack those B cells that's producing the cytokines and the autoantibodies. So the first step is to acquire the T cells out of a blood sample. So this can occur either autogenic, meaning self, meaning that the person who is going to get the CAR T cells in the end are the ones that donate the blood to get the T cells out in the first place, versus allogenic, meaning that it comes from a different person. But regardless of how the cells are collected, they are isolated from the blood. And then the next step you can see is that there is a gene that is inserted into the cells that manipulates what the receptor on the cell surface looks like. So you can see the turquoise receptors on the cell surface have been changed because of the DNA that's been inserted inside of that cell. And then once that happens, the goal is to grow as many of these cells as possible. So when they're now given into a patient, those cells are active and ready to go. And their goal is to attack and find those B cells to prevent them from causing further damage. So as I mentioned, the purpose of CAR T cell therapy would be to manipulate T cells so that they drive and they attack autoimmune diseases. And there have been several human studies that's taken place thus far that have used both the autogenic, which are the self CAR T cells, and the allogenic, which are the other CAR T cells. And they've been shown time and time again in several different patients that have improved lung, skin, and heart function in the patients that had received these CAR T cells. And there are currently a number of clinical trials that are going on right now to further study CAR T cells in scleroderma. In addition to CAR T cells, there is something that's called CAR NK cells. So think back to that picture that I showed you before, and we talked about those innate cells that's a part of the immune system. The NK cells, or the natural killer cells, are those innate cells that are also very important for the immune system to function. And there have been other researchers to find that you can even manipulate these natural killer cells as well, just as you can manipulate the T cells to create CAR NK cells. Again, they're a type of immune cell that's important in fighting infections, and they can be manipulated to attack B cells. And so there are also clinical trials that are currently undergoing and in the works to see if natural killer CAR T cells would be, natural killer CAR cells would be important as well. And then finally, there is another manipulation that can occur called BITE cells. So BITE stands for bispecific T cell engagers. So you can see that this purple and blue connector that's present, it is made up of a part of two antibodies that are connected together. So one part of the protein recognizes B cells, and the other part recognizes T cells. And so you can insert them on the top of the cells, and it brings together the B cells and the T cells as shown in the other picture to your right. And you can see that when the T cell and the B cells engage, the T cells become activated and they can release proteins that can then attack the B cells. And so it's used as a different type of way to, again, activate the T cells to then attack the cells that are problematic and causing the disease. This has not been used in scleroderma thus far. It's been used in several different types of cancers, but there are already researchers kind of thinking that this may even be another step or another mechanism that might be beneficial in scleroderma. So we've talked a lot about these cell-based methods to get rid of the B cells that causes the autoantibodies and contributes to the inflammatory proteins. But there's another way to delete B cells. And when Dr. Hashimi was here, she was talking about rituximab, which most of you may have heard of, which is a medication that is targeted against B cells. However, there is another medication that's just been approved or looks as if it will be approved for lupus patients that's called obinutuzumab, which is another way to target B cells. So again, if you look here, the little Y-shaped structure again is an antibody. And obinutuzumab is an antibody that can recognize the surface of B cells. So the CD20 is kind of the address to say, hey, I'm a B cell. And obinutuzumab can recognize that cell surface marker on a B cell and then cause the B cell to kind of self-destruct, again, as another method and way to get rid of B cells. And I say all that to say because there was a mouse study that was interested in looking at if we combine these two different methods of getting rid of B cells, will we have an even better outcome? Almost like it's two better than one. So in this study, what they did is they combined using CAR T cells as well as one of these medications to get rid of B cells, and they introduced it into mice and they wanted to see if they could have an improved outcome. These mice were chemically treated, so they developed something like scleroderma. There's no mouse model that really captures everything that happens in a scleroderma patient, but sometimes these studies can be helpful in the beginning. And so these mice that were clinically treated to develop a scleroderma-like disease were given these two modes of getting rid of B cells. And unfortunately, what they saw is that the mice developed like worse lung disease as well as worsening of the pulmonary hypertension, which sounds negative. And obviously that was not the intended purpose of the study. But I say all that to say that not every study comes out with the best outcomes, but in every single study we learn something. So there was always something to learn even from this study that seemed as if it was a negative outcome. So as we go forward and we think more about these therapies and how we can incorporate them in patients, these are studies that are also very beneficial and important for us. So another way outside of manipulating cells to get rid of B cells and other problematic factors in the immune system, or using medications to get rid of problematic cells, is by using transplantation. And so some of you may be aware that stem cell transplantation was very important and transformational for a lot of our patients with scleroderma. And what happened there is blood stem cells were isolated from the person, and then their immune system was deleted using medication, and those previously isolated stem cells were then given back to the patient to restart their immune system. And many patients had great outcomes from that. However, those studies and those treatments have tended to be reserved for the more severe patients. So we've been looking at other mechanisms that could possibly be used to isolate stem cells from other sources. So there is something called mesenchymal stromal cells. And these are another source of stem cells. So based on this picture, you can see that you can actually get these stem cells from lots of different sources. So they can be isolated from the bone marrow, but they can also be isolated from adipose tissue, which is fat cells. And it can also be isolated from neonatal tissue like umbilical cord tissue. And when these cells are isolated from these different sources and you get the mesenchymal stem cells, it's been shown in several diseases that it could treat liver disease, it can treat kidney disease, cardiovascular disease, and there's been studies recently to see it's beneficial for lupus patients. So we're now looking to see if this could also be beneficial for scleroderma patients. So again, mesenchymal stem cells are adult stem cells that can be isolated from many organs, and they can release different proteins and factors that work to suppress the immune system so that it's not as activated, and they can also work to decrease fibrosis. And these studies have mainly been done in cells, in cell cultures, kind of cells growing in a laboratory. There have been some animal studies also to show that this might be promising. And there have been some small studies in humans also to show that this actually may be a promising therapeutic. So scattered throughout my presentation today, you'll see kind of where I highlight how MUSC has been contributing to some of these advances in research. So Dr. Gary Gilkison is a researcher here at MUSC who's been focused in lupus and has been really focused on looking at these mesenchymal stem cells and see if they could be beneficial in that instance. Dr. Marie Hudson is from Montreal, Canada, and she is currently working on a clinical trial using human umbilical cord mesenchymal stem cells in the treatment of scleroderma. And Dr. Gilkison here at MUSC was the one who provided her the source of the umbilical cord stem cells. So we're really excited and grateful for the collaboration that occurred, and we're anxious to see what the results of the studies will be. Still on the same vein of looking at inflammatory proteins and cytokines, we want to see if we could target some of those cytokines that's known to be increased in patients with scleroderma. And this is not necessarily a new study, but this is broadening some of the other cytokines and other pathways that we could try to target. So we already know about Actemra or tocilizumab that's been FDA approved to treat scleroderma lung disease. And the way that that medication works is that it is an antibody. Remember those Y-shaped structures I showed you before that blocks the IL-6 receptor. And IL-6 is one of those cytokines that's known to be elevated in patients with scleroderma. Tofacitinib, which is also Xeljanz, which is a JAK inhibitor, which you heard earlier today from Dr. Hashimi, has also been one of those newer therapeutics, like I'm loud, one of those newer therapeutics can also work to block IL-6 from signaling inside of the cell. So you already heard that it has been used in sparingly kind of throughout the university. And the hope is that one day we'll have FDA approval that this can be more widely used in scleroderma. There is another pro-inflammatory molecule called interferon. Interferon is also known to be elevated in patients with scleroderma and is currently FDA approved for treatment in lupus. And anifrolumab is also an antibody that blocks the interferon receptor from signaling inside of the cell. So there's currently a clinical trial that's going on right now called DAISY. And the point of DAISY is to treat scleroderma patients with anifrolumab to look for benefits of using this medication for possible treatments. And in this trial, it's targeting and looking at both patients that have limited scleroderma as well as diffuse scleroderma. So we've talked about kind of a wide range of different approaches and how we could possibly treat inflammation in our patients. So now we're going to switch gears and we're going to talk about pulmonary arterial hypertension. So again, this is another MUSC collaboration and connection showing how we are constantly looking for better ways to treat pulmonary arterial hypertension. And this is a collaboration with Dr. Raul Argula, and Dr. Friedman, who are in the pulmonary department, and our very own Dr. Fagali Bostwick. And the goal of this study was to look for differences in patients with and without scleroderma that have pulmonary hypertension. So in this study, what they did is they were able to get lung tissue from patients with pulmonary hypertension, again, with and without scleroderma, and look for differences in the tissues of these patients. And what they found is that patients that had scleroderma and had pulmonary arterial hypertension actually had more inflammation of the lungs. Again, going back to those B cells and those T cells that we talked about earlier. So again, showing that possibly depleting B cells and T cells might be very helpful in the treatment of pulmonary arterial hypertension for patients that have scleroderma. There was another study that was published in 2024 that was also focused on pulmonary hypertension. And in this study, instead of looking at the actual lung sections and looking for changes, this team decided to look at the blood of patients with scleroderma and those patients that had risk of developing pulmonary arterial hypertension. And what they found is there was over 200 different proteins in the blood samples of patients that had scleroderma that were at risk for developing pulmonary hypertension. And the majority of those proteins were actually inflammatory proteins. And when they looked closer at the patients that had scleroderma to see if they could figure out pathways that these proteins belong to, most of the pathways that seem to be important in these proteins or directly related to these proteins were IL-6. So again, remember IL-6 that we talked about, and then another inflammatory protein called IL-17A. And when they took skin cells of patients that had scleroderma and they blocked IL-6 and IL-17 using different mechanisms, they found that there were certain proteins that decreased in those skin cells that were actually also important for the development of pulmonary hypertension. So they concluded that possibly using medications that block both IL-6 and IL-17 may be a future potential treatment option for pulmonary hypertension in scleroderma. Now we'll move on to talking about gut health, and we get lots of questions and lots of conversation about kind of how does diet kind of impact kind of disease pathogenesis. So we'll start with the microbiome. So what is the microbiome? The microbiome is a collection of different microbes. And so a microbe could be bacteria, it could be fungus, it could be viruses, but they are microbes that live naturally either on our surface of our skin or inside of our bodies. And in the gut, so when we're talking about the gut, we're talking about our stomach, we're talking about our intestines, we're talking about our colon. The microbiome that exists within those organs may influence the development of autoimmune diseases. And so there were other studies to show that depending on the makeup of what the microbiome is inside of our gut, it may exacerbate the development of autoantibodies, and it could cause inflammation to happen. There were others who looked at the differences in the gut bacteria that's seen in patients that have severe scleroderma and in healthy donors. And they did see that there was a difference in the type of bacteria that was present with patients that did, or donors that did not have scleroderma and patients that did have scleroderma. And it's also been shown that the gut microbiome, the ones that's inside of us, can communicate with other organs. So based on what we come in contact with in our environment, like the type of antibiotics that we may take, other medications that we may take, pollution, as well as the type of foods that we eat, can all impact the type of bacteria that lives inside of our gut. And the way that they can signal to other organs is by releasing different types of chemicals that can interact with adipose tissue. It can interact with our liver, it can interact with our brain, it can interact with our cardiovascular system, and it can interact with our lungs. And there were several studies to show this. But the question is, does those microbiome signals, does it impact anything in scleroderma? So there was a study to look at that relationship to see if the bacteria that's inside of our gut can affect the skin bacteria or communicate with the skin microbiome that's present. And when they looked at the differences with the skin bacteria and the gut bacteria, they did see that there were differences, but those differences were actually dependent upon the type of autoantibody that the patient has. So if the person had the autoantibody that was positive for the centromere, they had certain types of gut microbacteria. And for those that were SCL70 positive, they had other different types of gut microbacteria. And they did find that looking closer at those types of bacteria, kind of the nitty-gritty of the actual names of those bacteria, could impact the skin score. And remember, the skin score is how we determine the amount of tightening that's happening in the skin. And the type of bacteria that's found on the skin of patients with scleroderma has been identified in other inflammatory conditions like psoriasis, which is also a very inflammatory autoimmune disease in the skin. So some of those bacteria were actually common between the two conditions. So this study doesn't give us more information about what types of food that we eat or what we should do, but it does lay the groundwork that it's possible that these things are impactful in the disease process, and we need further information and further research to try to make further stronger connections. So the next topic that we're going to talk about is fibrosis. And under that topic, we want to talk about the genes that are associated with fibrosis, as well as possible inhibitors for fibrosis. So again, we have another MUSC collaboration between Dr. Fagali Bostwick and Dr. Betty Sau, who is a researcher also in the Division of Rheumatology, who has done fabulous research regarding lupus. And she was looking at a gene called NCF1. And with that gene, she found that when that gene is not functioning properly, it increases the risk of people developing certain autoimmune diseases. And so she's looked at lupus or systemic lupus erythematosus. She's looked at another autoimmune disease called Sjogren's syndrome. And she also looked at rheumatoid arthritis. But in this research study, she also found a very similar association in patients with diffuse scleroderma, as well as in lung fibrosis. And when they look closer at how this gene functions in mice, they did see that manipulation of these genes in mice did affect lung inflammation and inflammatory cells. So, we always ask the question kind of where did this disease come from and kind of what was the origin of it? So these studies are very, very important as we're looking at genes that may be important and factors as why scleroderma may have developed, or they could be potential treatment targets in the future if we find that it is something that's prevalent throughout many patients with scleroderma. There was another study that was looking at a chemical that could be a possible inhibitor of fibrosis. So this protein, the way that it possibly could inhibit fibrosis is that it prevents the fibrotic proteins from coming out of the cell. So the way that fibrosis tends to take place is that the proteins are made inside of the cell, and then there's different mechanisms to allow the proteins to come out. And the way that this inhibitor works is that it blocks the cell from releasing those proteins. And so they tested how this inhibitor would work in skin cells. They were scleroderma patient donors. They grew out skin cells from there, but they also collected donor cells from people without scleroderma, and they were treated with different chemicals that could allow fibrosis to take place. And the results were that when treated with this particular inhibitor, it decreased the amount of collagens and fibronectins, which are the fibrotic proteins that we tend to think of. It decreased the amount of those proteins that were released from cells. And importantly, when the cells were treated with this inhibitor, it did not cause cell death, which is great because obviously if we want to continue on with this inhibitor, causing cell death would be problematic. And so it shows that it's not toxic to our cells, which is very, very important. And to limit the potential side effects that could possibly happen if this potential inhibitor goes forward is that it only inhibits a specific portion of the cell. So it doesn't inhibit widespread areas in the cell, again, which is important because we don't want other unaffected cells to be affected by this therapy. And then finally, they found that this inhibitor possibly could act in a local area, which is also important because you could think that this could be used as a topical thing. So it could be something that could just be rubbed in on the skin if this goes forward. So this study was very important because not only did it define a potential inhibitor that could be used in the future, but it also showed that they started thinking about how toxic these things could be to people, or even how this could be used as a potential therapeutic, the route that it could be used as a potential therapeutic. So then finally, we want to talk about social determinants of health and mental health. So I feel that as time has been going on, and as we've been learning more about scleroderma, we understand that we can't just think about it from a medication point of view, but we have to think in totality of how we want to treat patients so that we can get a holistic view of what's happening with us. So scleroderma affects the health-related quality of life. So what is the health-related quality of life? It's defined as the influence of the disease and treatment on how a person functions and their overall sense of satisfaction. And so that's really, really important when we're talking about having, this type of disease. I don't think I need to tell you that. And so there are definitely issues that affect the health quality of life. So the disease itself can affect it. So for instance, I know several patients, we talk in clinic about the fatigue that comes as a result of having scleroderma. And so obviously this is problematic because it affects one's ability to care for themselves and to engage in different activities. So then that could decrease the health-related quality of life. But there's also psychological distress that can also occur, especially for people who may have underlying mental health issues before even the development of scleroderma, most prevalent anxiety and depression. And then there's other social determinants of health that could then also factor into these psychological issues. And so if we don't address all of these different layers, it can lead to ineffective disease management. So our goal is to identify, discuss, and incorporate mental health issues into the care of individuals with scleroderma. And the author of this study and everyone, I think, would agree that this is very important. So when we're talking about social determinants of health, it's defined as the social and economic conditions where people live, work, learn, and play. And the presence or lack of certain resources can not only impact health, it is what we call a factor in the social determinants of health. For instance, higher healthcare costs is a social determinant of health. And so if someone has higher healthcare costs, then they're not able to seek the treatment that they need, and it can also lead to psychological distress, as well as food insecurity. If you have to choose between taking your medications or being able to afford groceries, obviously that can negatively impact your mental health, your physical health, and is a factor in social determinants of health. So all of these things can lead to psychological and social stress. And there are specific social determinants of health that patients with scleroderma may experience. So because these psychological distress can impact disease as a whole, it has to be addressed. However, in the clinical setting, when you're before your physician, sometimes time is very limited. And since we're now recognizing these issues, we still have much more research that has to be done so that we can find the proper ways to find solutions for this. But one of the things that the author recognizes is to encourage self or patient-focused solutions. And what that means is to encourage us to work through our emotions. This will allow those anxious feelings to hopefully dissipate over time. It will help to grow self-awareness and emotional strength. And it will also lead to feeling more empowered and not being so isolated. Also participation in support groups. So shout out to our support groups because they're important because it allows us to gain access to more information and education. It gives more knowledge and power, and you can see how that could feed forward to growth in self-awareness and emotional strength. It leads to feeling more empowered and again, decreasing those feelings of isolation. So this was a diagram that was included in the article by this author. And these are different recommendations for self-care. And one of the things that I wanted to highlight that she talked more about was writing down questions to discuss with your provider. And while doing that at home, you can also develop a goal of care plan. And so this is meant to be completed at home and then brought to visits. And so in each one of these subsets, it makes you kind of think a little bit closer about what's happening with you and what you would like the outcomes to be to start a conversation with your provider. So looking at the concerns and the barriers that you experience. So one example says, the main things I would like to improve my health care. symptom management, meaning the main symptoms I wish to reduce or eliminate. What are your treatment goals, and kind of outlining what those are, and then outlining what you think the expected outcomes is, again, to start a conversation so that both of you can agree on what should be done and what can be done going forward. So again, our MUSC connection is with Dr. Sarah Smith. She's a future rheumatologist. She completed medical school here and residency at MUSC, and she's on her way to Johns Hopkins University to complete her rheumatology fellowship. While she was here at MUSC, however, she also completed a master's degree, which focused on social determinants of health and scleroderma. And the title of her thesis is Exploring Social Determinants of Health, Health-Related Quality of Life, and Clinical Outcomes in Scleroderma. And she has been a long volunteer and organizer for the conference since she's in medical school, and she is the proud mom of Robin. So what does the future look like as we're talking about treatment development? Again, here at MUSC, we are proud to say that we have 3 laboratories that are actively producing and in the process of developing drugs that have the potential of decreasing fibrosis. So we have Dr. Stan Hoffman with his company called Fibrotherapeutics. We have Dr. Silver and Dr. Bogatovich with their company Fibrobiologics. And we have Dr. Fagali Bostwick with her company Fender Therapeutics. So again, we are actively standing by research and hopefully making a difference for our patients. So in conclusion, I hope I have convinced you that this is a very exciting time for scleroderma research nationally, and MUSC is one of those national leaders for scleroderma research, as well as internationally. I hope I provided a snapshot of research topics, but obviously much more research is needed. We need to understand the safety and the long-term effects of the potential therapeutics that I talked about. And we also need to understand and develop better resources to help with issues related to mental health. So more to come in 2025. Thank you, and I'm happy to take any questions.